Well, I want to talk about bioluminescence and why an organism would expend a lot of its energy for light production. But before I do that, I want to talk a little bit about the actual mechanism of light production. So how do organisms actually produce light? Well, they use a chemical compound that is called a luciferin, and the luciferin is a chemical. It's not any one chemical. It is per species, but there are many kinds of luciferin, and it usually uses an enzyme called a luciferase. It uses ATP as an energy source. It may use ions like magnesium to catalyze the reaction, and it uses molecular oxygen from the air to carry out the reaction. So basically, that's the short explanation of how light is being produced. But what's interesting is, unlike other processes that have occurred through evolution, let's say like the development of an eye, for instance. If a primitive organism developed an eye, then that eye would be elaborated on by other organisms that through evolution, other organisms would take that eye and probably improve upon it. Well, that's not so with bioluminescence. In the case of bioluminescence, it's as though every time a new organism came along that was going to be bioluminescent, bioluminescence had to be reinvented. It was as if God had to create bioluminescence de novo. It's like God saw the light and it wasn't so good, or maybe it was good, but it wasn't the right kind of light. And in fact, what's really going on here is that these organisms, they all live in somewhat different environments. They have different nutritional requirements, and they all produce different biological compounds. So it may be easier for one organism to produce a particular kind of luciferin than it is for another one. So I want to show you some of the actual chemical structures of luciferins, and we'll see some similarities and differences between them. And I've drawn this on this board here. So I want to show you two types right up at the top. The first one is a compound called solanterazine, and this is something that you will find in sea combs. Many sea combs, ctenophores are bioluminescent. There are also a number of cnidarians that are bioluminescent. So you have both things like jellyfish that are bioluminescent and the sea combs, and they use this kind of a structure. What you have here is a 6-membered ring with 2 nitrogens in it, and then a 5-membered ring fused together with that, and this is called a parazinoimidazolone, this type of ring structure. And you'll see it in solanterazine, the compound that I show you here, which is one of the luciferins for the selenurids, the jellyfish group. And then you'll see it over here again, another compound called vargulin, which actually has the same ring system. Here it is again, the 6-membered ring and the 5-membered ring. And vargulin luciferin is found in vargula. Vargula is actually a form of ostracod, and the ostracod is it's a kind of it's a very small mollusk type of creature, but this is typical of the luciferin that you would find in this creature. So there are two that are fairly similar, but then I want to show you another one here that has no structural similarity whatsoever. This is found in Diplocardia. Diplocardia is actually a terrestrial earthworm, a particular kind of earthworm that gives off a bioluminescent glow. And what's characteristic about this is it has an aldehyde function. Let me put this down here and show you again. So you see this C double bond OH here. That's what we call an aldehyde. And I want to show it to you again in compound 4. which is a form of bacterial luciferin, and this is called tetradecanal. It's a very simple molecule. It's only got carbons and hydrogens, so it's a 14 carbon chain, and the terminal carbon, instead of having 2 hydrogens, has an oxygen on it. And this is typical of the kind of luciferin that you would find in a lot of bacteria. Then there is coleopteran luciferin, and that's shown over here. And this has 2 rings that have a nitrogen and a sulfur in it, also a benzene ring fused to that. So this is a benzothiazole, and this is another thiazole over here. And this is actually found in the firefly, in the terrestrial firefly. So unlike the other ones that I showed you before, which give off blue light, blue being the light that is most easily transmitted in the water, what we've got here is something that gives off yellow light. Here's another compound, again much different than the other ones. This is an ester of formic acid, and it also has a 6-membered ring with a double bond in it here. And this is Latia luciferin, and Latia it's actually a limpet, again a mollusk, but again entirely different kind of structure. And finally, the dinoflagellates that I was talking about before, they use a much different luciferin than the other kinds of molecules. And here's the dinoflagellate luciferin shown right here. Let's see if I can get a better focus on this. Here we are. And what I saw this structure, I immediately recognized that this is a derivative of a compound called porphyrin. So you see you've got 4 5-membered rings with nitrogens in them. Now normally in porphyrins, these are connected, all these rings are connected together. Here you'll notice that there's actually a break in the rings right here, over here. But if this carbon atom were actually connected through here, this would be a porphyrin-like structure. If it had magnesium in it, it would be the porphyrin, it would be similar to the porphyrin that makes up chlorophyll. If it had a iron in here, it would be similar to the porphyrin that makes up heme, which is what transports oxygen through blood. But these organisms actually have chlorophyll in them, and they can actually use the chlorophyll to make the luciferin. So they don't have to design luciferin from scratch. They can take some of their chlorophyll and convert it into the luciferin. And what's interesting again about these things is I'm going to show you the solanterazine again, and I want you to see how this actually reacts with oxygen. What it does is it produces a highly reactive species, which is called a dioxetane-one. I'm going to show you the basic structure of dioxetane. It's just a 4-carbon ring, a 4-membered ring with 2 carbon atoms and 2 oxygen atoms. The one that I've shown down here, which also has 2 carbonyls, that's a double bond to an oxygen coming off of this, this is called 1,2-dioxetane-dione, and it's actually the substance that is found in these light sticks, which when you break them open and shake them, you go to somebody's bar or bas mitzvah, and they usually have these at parties. They give these things out as favors. The light that's being produced is because of this particular molecule, but it's the same molecule that are very, very similar to the molecule that is used to produce light in the solanterazine oxidation. So that's something that is characteristic, that particular. I also wanted to show you here, even though I don't show it, the land animal, the firefly, also goes through a dioxetane intermediate for light production. So that's kind of an interesting observation. In any event, I want to talk finally about why an organism, that's how light is being produced,